Hi, all of you. Today, let's learn about general principles of microbial pathogenesis. So, first, introduction to microbial pathogenesis. You need to know what is microbial pathogenesis. So, whenever you are affected with some microbe, when a microbe or any microbial organism like your bacteria, fungus, virus enters your body, what happens? Yes, so that is what is about this chapter is about. Microbial pathogenesis that refers to mechanisms by which microorganisms can cause disease in a host, right? So how does it cause a disease in you is what is the pathogenesis. This involves the interaction between the pathogen and the host immune system. So when a bacteria enters the bloodstream or when a bacteria enters your body, what happens? Your immune system, your body's own immune system will start reacting to the pathogen, right? So what is the interaction about? And how does this interaction bring about a disease is what we are going to learn. So here in whole of this chapter, you're going to understand about different terminologies that you use here. Colonization versus infection versus disease. So what is colonization? The presence of microbes in the host without causing a disease. So just the microbe has entered the body. Yes, and it has started to find its place. Yes, suppose it is a lung infection, it has entered your body through your respiratory tract, and it has colonized in the lungs. If it is an intestinal infection, it has colonized in your intestine. So first it will go and find its place. After that, it is going to multiply. After that, it is going to invade your organs and then it is going to cause disease. So at the point of colonization, there is no symptom. So there is no disease that has happened. Next is infection, the invasion and multiplication of a pathogen in the host. So how does the invasion happens and how does the microbe, how does the bacteria gets multiplied in the host? Next is disease. So after which what happens? The disease is going to happen, the damage is going to happen to the host, right? So this is what is these three terminologies about. What is a normal flora whereas a pathogen? Pathogen means the bacteria, virus, fungus that is going to cause diseases. What is a pathogen? Normal flora means what? In all of your body. Yes. So like your skin, your gut, your intestine, in all these areas, there will be something called as normal flora that are non-pathogenic. So they are not going to cause disease. They will exist along with, they coexist with the host, which means that it is not going to cause disease. Yes, sometimes it will be useful. Yes, or useful also, like your gut microbiota is going to be useful to you, right? So sometimes it is harmless. Yes, it doesn't do anything. So those are all normal flora. They are not going to cause disease, whereas pathogens or organisms that are going to cause disease. Next is the type of my pathogenic microorganisms, bacteria, viruses, fungi, parasites, and prions. Bacteria are all these are the examples. The common ones are Staphylococcus aureus, Mycobacterium, TB. Viruses like HIV and influenza virus, fungi, candida, and aspergillus. Parasites like your plasmodium and entamoeba. And prions, which causes the disease. These are the proteins. After infection happens, no, these are the proteins that are part of your pathogens that is going to cause disease. It's going to get accumulated and it is going to cause disease. So what disease can prions cause is a Creutzfeldt-Jakob disease. Next is host microbe interaction. So how does your body and your bacteria or virus or any pathogen going to interact? One is symbiotic relationship. So that is commensalism, mutualism, parasitism. Commensalism means what? The microbe benefits, but host is neither helped nor harmed, right? So here what happens? The microbe is alone living. Yes, it is taking the nutrition or it is taking the place, but the body is not going to be helped or harmed also. It's nothing. So that is what's commensalism. Mutualism means what? Both the host and the microbe will benefit from each other. Parasitism is the microbe alone benefits at the expense of the host. So it is going to cause disease in the host, right? But the microbe will eat away all the nutrients and it is going to cause disease in the human body. So that is what is parasitism. Next is opportunistic versus obligate pathogens. What is an opportunistic pathogen? For example, HIV infection. In HIV infection, what happens? The immune system, the immunity of the host is going to be decreased. So immune system will be weakened. So due to weakened immune system, if some pathogen is going to get multiplied and it is going to cause disease, that is what is called as opportunistic pathogens. So opportunistic pathogens will cause disease when the immune system is weakened, right? For example, it's going to be in immunocompromised patients, pseudomonas is going to cause disease. Obligate pathogens means whatever it is going to cause disease, whether your immunity is strong or weak. When the amount of the bacteria, the amount of quantity of bacteria virus is going to uh, go beyond a certain level, then it will cause disease. Okay, it's definitely going to cause disease. That is what is Mycobacterium TB, right? So this is one example. Next is host susceptibility factors like your age. So in the newborns or old age, they are more susceptible to infections. Immune status, reduced immune status, they are more prone for infections. Genetics, in certain genetical factors, there will be immunocompromised state. So in that cases, there will be more prone for infections. Comorbidities for patients who have diabetes. Comorbidity. So when a patient has diabetes mellitus, he is more prone for developing infections. Likewise, environmental factors, right? So these are different factors that make people more prone for developing infections. So this is about your host microbe interactions. The next, the next is pathogenicity and virulence. So what is pathogenicity? The ability of a microorganism to cause disease is what is pathogenicity. So how effectively the microorganism is going to cause disease. That is pathogenicity. Virulence means what? Degree of pathogenicity. Yes, that is measured by lethal dose 50% or infectious dose 50%. So how much dose of the virus is necessary? How many viruses will be necessary to cause infection in 50% of the population? So that is what is infectious dose, and how much is necessary to cause death in 50% of population is going to be lethal dose 50. Okay, so this is the degree of pathogenicity. So virulence is nothing but your degree of pathogenicity. Then virulence factors. Virulence factors will help in colonization, immune evasion, and host tissue damage. So how effective your virus is, how strong your virus is, how infectious your virus is or bacteria is, that helps in. So sometimes the bacteria will colonize so fast and it multiplies so fast, right? And it will start evading your immune system. So it will destroy all your T cells, it will destroy all your B cells. Yes, so it will destroy your T cells and B cells, and it will reduce your immune system capacity and cause tissue damage. It will also cause tissue damage. Next is mechanism of microbial pathogenesis. So we will see various mechanisms of microbial pathogenesis. So first one is adherence. Adherence means what? Attaching. Okay, how does your pathogen attach to the host tissues? How does it attach? With the help of villi or fimbriae or adhesins or biofilms. So look at the picture here. This is a villi, right? So they are hair-like projections out of your bacteria. So fimbriae also the same thing. So similar to this. So villi and fimbriae or these are finger-like projections or hair-like projections with which it is going to attach to your attach to your tissue, right? So example is E. coli. Similarly, adhesins. So adhesins, which will again attach the bacteria to the host tissue. Example is Streptococcus pyogenes. Then biofilms. Example is Pseudomonas aeruginosa. Biofilms means what? So suppose look at this picture. First one is there is the bacteria. Yes. So this bacteria will start producing one layer on it. So inside this inside this bag-like thing, inside this film-like thing, it is going to multiply, right? And after it becomes mature, this film is going to burst and so much of bacteria is going to come out and again is going to cause it will go and colonize in another place and it does likewise the infection is going to spread, right? So this is what is a biofilm. So this is a mechanism by which adherence is going to take place. Next is invasion. So how does the bacteria attach to any organ? That is what is adherence and how it is going to invade. It is going to get inside this host tissue and it is going to cause disease, right? Because it should get inside your cells. It should get inside the tissues, right? So that is what is invasion. So how does it do? Certain pathogens will produce enzymes that is going to break down your host tissues. Likewise, it will create a pathway. Okay, it will create a pathway by breaking down your host tissues with the help of some enzymes and it will enter inside the cell. For example, hyaluronidase. Hyaluronidase, anything that is ending with ase is going to be a enzyme, right? So the pathogen is going to produce hyaluronidase that will degrade hyaluronic acid. Hyaluronic acid will be a connective tissue component. So that is going to be present in any tissue. So when it breaks this hyaluronic acid, what happens? The cells all will become loosened. The tissue will become loose and it will give way. Okay, so there will be cracks or it will give way for the pathogens to enter. Then collagenase that breaks down collagen. So that also gives you the strength to any structure. So if that strength is broken down with the help of this collagenase, the enzyme and the pathogen is going to enter into your tissues. Next is coagulase that promotes clot formation to protect your bacteria. Coagulase, as the name says, yes, coagulation that promotes clot formation and that will protect your bacteria. So the bacteria will not be broken down by your immune system. So in turn, the bacteria is going to live for long, it is going to multiply, right? Next is kinases. Kinases will break down clots to facilitate spread. Kinases will break down clots and that will help in spread of bacteria. This is about invasion. Next, what happens? Evasion of host defenses, right? So how does the bacteria or virus or anything is going to escape your host defense? So initially, when a bacteria is going to enter your body, your immune systems will start attacking, right? Your immune cells is going to attack the bacteria. But how does some bacteria will have some structures which can protect itself from the body's immune system? Okay, so how does it do that? That is what is evasion of host defenses. One is with the help of capsules. Capsules, as you can see here in this picture, there is something that's surrounding the bacteria, right? So that is what is capsule. So with the help of this capsule, your, that can prevent the phagocytosis. Phagocytosis means what? The cell eating. So the macrophages or the WBCs in your immune system is going to eat up the bacteria. With thinking it is a foreign body, it is going to eat up the bacteria. That process is what is phagocytosis. So capsules will prevent the phagocytosis. So that is thereby it will evade, thereby it will escape the body's immune system. That is the example is Streptococcus pneumoniae. Likewise, antigenic variation. Antigenic variation means what? On the surface of the bacteria, there is going to be some antigens, right? This antigens, to this antigens, the body is going to produce antibody, right? So what happens when the bacteria or virus is going to change its surface antigens? Yes. So while it is getting multiplied, it is going to change its surface antigen so that it will escape the immune detection or escape the attack by immune system. Example, Neisseria gonorrhea. This bacteria is capable of changing the surface antigens. Next one is intracellular survival. Some pathogens will go inside the cell and it will survive inside the host cell. Okay, it will survive inside the host cell. That is what is Mycobacterium tuberculosis, right? So these are the mechanisms by which you can escape the host defenses. The next is So till now, what have we seen? We have seen about what is the mechanism. First one is adherence. It is going to attach to the host tissues. Second one is invasion. It is going to create some breaks in the host tissue so that the microbes can get inside the tissues. Next is evasion of host defenses. So when the host immune system is going to attack the bacteria will have some mechanisms to protect itself from the host's immune system. The next one is toxin production. So how does it cause disease? One is there are two types of toxins. One is exotoxin, the other one is endotoxin. Exotoxin means what? This is the bacteria. The bacteria will produce some substance. These substances will cause disease. Okay, that is exotoxin. So exotoxins are nothing but secreted proteins that will damage your body's cells. Okay, one is AB toxin. Yes, so there will be A toxin, B toxin. So one toxin will help an attachment of the cell to the, like for example, an organ like intestine. The other toxin is going to be pathogenic. It will cause disease. So these kind of actions it will have. So this is A toxin, AB toxins. Like for example, Corynebacterium diphtheriae. This bacteria is going to produce diphtheria toxin. Then you have cytotoxins. These are going to break. Cyto means cell. Okay, toxin. So this toxin is going to break some cells. So like hemolysis, for example, that will break the red blood cells. Next is neurotoxin. It can cause nerve damage. Botulinum toxin causing paralysis. This is an example. Enterotoxin, entero means intestine. Okay, so this toxin is going to cause diarrhea. Yes, that is cholera toxin. These are all exotoxins. So they are proteins that are produced from the bacteria or virus that is going to cause disease in your body's cells. Next is endotoxin. Endotoxin means what? Like, for example, is lipopolysaccharides from gram-negative bacteria like E. coli. So here, as you can see, you have so this is a structure that's found, okay, on the bacteria. So this is the endotoxin. So exotoxin is something that is secreted out of the cell. Endotoxin is something that is on the surface that is going to cause disease with the help of these antigens on the cell wall of your bacteria. Next, something called as super antigens. They will cause excessive immune activation, thereby causing disease, right? So that is what a super antigen. So next is stages of infection. First, what happens? It will enter. Bacteria or virus or anything will enter your body through any tract, like respiratory tract, gastrointestinal tract, or urogenital tract, or through any wounds. That first step is entry. After it enters, colonization. It gets adhered to your host cells. That is what is colonization. Next, what happens? The bacteria starts starts multiplying and it will invade your host cells. Then it will spread within the host. It will start spreading. How does it spread? It will spread via your blood or lymphatic system. Next, what happens? It will cause tissue damage and it will also cause immune response. So those are inflammations, or it can cause apoptosis, necrosis, or the bacteria that is producing toxin will cause toxin mediated damage. Then exit, transmission to new hosts, like for example, through respiratory droplets or fecal matter. How does it spread to person to person? Okay, so these are the different stages of infection. So you can just think about it, how a bacteria enters and how does it cause disease. That is the stages of infection. So just look at this picture. Source of infection through respiratory tract. It is going to enter into your lungs. It is going to get colonized in the lungs. It will multiply and it will start invading. And then it will enter into the bloodstream and it will go to various other tissues and it will affect all the other organs. Okay, so this is how an infection can spread. The modes of transmission. What is a mode of transmission? So from one person to another person, how is the infection transmitted? One is direct contact, person to person, like skin infections, right? And sexually transmitted diseases is person to person contact. Indirect contact through fomites. Yes, these are contaminated objects, like a person sneezing and he is he's keeping his handkerchief on some other place and that handkerchief is used by some other person. Yeah, so these are all indirect contact. Droplet transmission like coughing, sneezing, these are droplets through which your infections will spread. Airborne transmission through air. Yes, tuberculosis, measles. So these are the whenever small particles remain suspended, so everyone is there in some same room. Okay, and there are 2-3 persons who are affected, and they are coughing and sneezing, or they have some infection in them, and that's all airborne. Okay, that is going to transmit to other people in the particular room. That is airborne transmission. Vector-borne transmission, the vectors, there is vectors, some carriers involved. Like for example, malaria. Malaria is transmitted via mosquitoes. Dengue is transmitted via mosquitoes, right? Then fecal-oral route, contaminated food and water. Through that, like cholera, hepatitis A, these are all fecal-oral. So whenever you have contaminated food, contaminated water. These are the different modes of transmission. Next is role of host immune response. One is innate immunity is the first line of defense. Yes, all your skin barriers, right? Skin barriers, mucosal barriers, macrophages, natural killer cells, cytokines, these are cells inside, right? So these are first line of defense. So whenever some foreign body is entering inside, these things will actually help us in first place. Next is adaptive immunity. You have humoral and cell mediated. Cell mediated means T cells combat intracellular pathogens. Humoral immunity means B cells and the plasma cells are going to produce antibodies. Right, those antibodies are going to be produced against particular antigens or the particular bacteria, and thereby it is producing immunity. That is humoral immunity. Cell mediated means the T cells. You have the cytotoxic T cells, T helper cells that is going to kill your pathogens. Next is immune evasion strategies that we know the things can hide inside the host cells. Yes, some bacteria can hide inside the host cells, what we saw already, right? Then suppressing immune responses. So like your HIV will infect your T cells. So when T cells are infected, the immunity itself is suppressed. That's why in HIV infected people, the immune system is compromised. This is about the role of the host immune response. Next, microbial pathogenesis in clinical disease. Again, what are the terms? Acute infection, chronic infection. Acute means short term and it's severe. Chronic means it's long term and it is be persistent for a longer period of time. Localized and systemic. Localized means one area is alone affected. Like for example, skin abscess. So the infection is localized to in one particular area. It is localized. Systemic means it is spread throughout the body. So through the bloodstream, the entire body is affected through the blood. Infection is spread through the bloodstream, which means sepsis. Latent infection. Latent infection means what? The infection can be present for a longer period of time. It doesn't cause disease, but due to some reactivation, it is going to come out of the dormant phase. Okay, so that is, for example, herpes virus. So it can stay in some place, yes, and then in some like nerve root ganglion or something, and then upon reactivation, it can start causing disease and causing symptoms. Till then, it will remain dormant. Okay, that is what is a latent infection. Super infection means a secondary infection due to disruption of normal flora. So, for example, in your intestine, we have normal flora, right? Normal microbes. That's what we had seen already, right? So, when you give antibiotics, for example, I thought it was Clostridium difficile infection in a patient, and I've given there was an infection in a patient. He had been treated with antibiotics. So that the infection settles, right? But what happens is the normal flora, normal gut bacteria will also die because I'm giving antibiotics. So now what happens because of the reduction in the normal flora, the pathogenic gut flora will start increasing, and that is what is Clostridium difficile. So this is a secondary infection due to disruption of your normal flora. So this is a super infection, right? The next is antimicrobial resistance and pathogenesis. What are the mechanisms of resistance? So what do we see? If you are infected with a bacteria, you are giving an antibiotic to treat or to kill the bacteria. Now what happens? The bacteria becomes very smart and it will try to try to develop some resistant mechanisms to the antibiotic that you're giving, so that it will not, so even if you administer that antibiotic, it's not going to work for the bacteria, because the bacteria has developed something to develop the resistance against the antibiotic. So what are the mechanisms of resistance? One is enzymatic degradation. So one the beta lactamase. Okay, so since this is an yes, this is the thing that we antibiotic is going to target. So, what if the bacteria is going to remove that target? The antibiotic itself will not come and act, is it not? So, finally the antibiotic is going to fail. The bacteria is going to win and it is going to survive. It will not be killed by the antibiotic. So that is the one mechanism of resistance. One is altered target site. So it's if for example, an antibiotic is targeting a particular site. Yes, antibiotic is targeting a particular site, but it is causing disease by some other site. So, which means that antibiotic is not effective here. Next is efflux pumps removing antibiotics. So, whenever the antibiotic gets inside the cell, there will be efflux pumps that is going to remove the antibiotics also. Yes, once its action is done. What if there are so many efflux pumps that most of my antibiotics are effluxed out? Yes, the action is not going to happen. So that these are the mechanisms of resistance. From the bacterial point of view, there will be biofilms. So I had shown you the picture of biofilms. Whenever there is a biofilm, they are covering around the bacterial multiplied bacterial community. Those biofilms will be resistant to antibiotics. Antibiotics will not work against them. Then horizontal gene transfer. So what happens when there is a gene transfer that is happening between bacteria? That gene is nothing but it is having the resistance to any particular antibiotic. Yes, so the resistance itself is transferred from one bacteria to the other via gene. Like for example, through plasmids or transduction or conjugation. So what happens? The resistance itself is being transferred from one bacteria to another bacteria. Next, prevention and control of microbial infection. So, how do you control or how do you prevent? By vaccinating. Yes, so from your young newborn age, you would have been vaccinated. Then, infection control measures like your hand hygiene, a personal protective equipment whenever you're in an infectious environment, sterilization techniques, disinfection techniques whenever you're working in your hospitals, right? So you are taking these infection control measures. Antimicrobial stewardship. So how do you rationally use the antibiotics to prevent resistance? So just one tablet. So if there is a course of treatment, okay. So till you get rid of the infection or get rid of the symptoms, you should be taking the antibiotics. But what if you just stop in the middle? Only when you stop in the middle, the rest of the bacteria which has not been killed inside your body is going to develop resistance. And it can easily transmit the resistance to the next generation bacteria, and that will produce a resistant bacteria. Yes, so we have to effectively use the antibiotics whenever you are in an infection, and you have to effectively kill the infection. Okay, before you stop the antibiotic treatment. Next, public health interventions. So during quarantine, you would have, I mean, during COVID season, you would have heard about quarantine. Yes, you should isolate the people from others whenever you are exposed. Then the surveillance. So you must keep looking for symptoms. Then outbreak control. Whenever there is an outbreak, you have to take steps to prevent the spread to other people. So these are the prevention and control of microbial infections.